My name is Harmit Malik. I'm a professor and co-associate director in the Division of Basic Sciences at the Fred Hutchinson Cancer Research Center in Seattle. I'm an evolutionary geneticist by trade, but I dabble in a number of different topics from microbiology to speciation research.
In this episode, we spoke with Harmit Malik about how he got into biology, books that inspired future scientists, and the importance of simply showing up. We discussed the Malik's lab research on genetic conflicts involving very diverse players, including centromeres, mobile genetic elements, retroviruses, and of course the host's innate immune response. The Malik lab studies these in several different models, ranging from yeasts to primates. Thomas Lemberger joined the conversation to talk with Harmit about peer review, preprints, and EMBO's journal-agnostic platform for manuscript review, Review Commons. Welcome to the EMBO podcast. Harmit started out as a chemical engineering student at the Indian Institute of Technology in Bombay.
Yeah, thanks very much for having me, Tiago. Looking forward to this conversation. Just like many other important transitions in life, I think my transition from chemical engineering to biology was chaperoned by a really fantastic mentor that I had. I had the pleasure of interacting with a mentor during my time at IIT Bombay, where I was getting a degree in chemical engineering, but I was getting more and more interested in biology. And so I was introduced, thanks to a chemical engineering professor, to a new professor who had just started at the Institute, Professor K.K. Rao. And he, got me interested in molecular biology. He was teaching a class on introductory molecular biology, but I couldn't attend the class because I had a clash with one of my required classes in chemical engineering. So he totally offered, completely unsolicited, that if you come over in the afternoon, we can just chat, over coffee. What he didn't mention was he was a chain smoker at the time. So, in a 1-hour conversation, I had the equivalent of like 1 pack of cigarettes just sitting across from him. But it was totally worth it, whatever damage it did to my lungs, because he was just a fantastic teacher. I think even after all these years, I've really not encountered anybody who had just this extremely seductive quality. He made even dry topics seem very interesting, but actually when he was teaching about the lac operon and the basic fundamentals of molecular biology, I was completely transfixed. And it became something that I felt like I could not pass up the opportunity to see whether this is something that I could actually spend the rest of my career in.
So this was not one, but multiple sessions, if I get it right.
This was actually not, this was over the course of 2 semesters. So I would say roughly around 60 hours of completely, voluntary work. I was not his student. I just want to remind you, I was not in his department. I was not his student. So he was just doing this. And then many years later, I think, I kind of went back, on his retirement party. I went back and I had an occasion to really talk to him, because I'm interested in what makes good mentors too, right? And I've had this experience of many good mentors in my life, and he was certainly one of the most influential. And I asked him, why did you do that? I mean, recognizing now the number of time pressures that a typical academic had, he just said that, yes, it's true. I didn't think about like how much of a time commitment would be, but I was pretty certain that you would give up. Like I've had other students who came and then, after a couple of hours, they stopped showing up because, you weren't doing this as part of a course requirement and stuff. So I figured that you will come for a few days and then you'll see that, okay, this is a lot of work. I'm going to stop coming in. But you never stopped. So I felt like I couldn't stop because like I had basically like encouraged you to do this. And so really that just emphasizes the importance of simply showing up, when you're supposed to. And that can actually make a pretty important influence on your mentor and basically motivate them to kind of rise up to be a good mentor for you as well.
He didn't try switching to cigars at any point to see if he could get some peace?
No. And, honestly, I think that, he was an academic in India and, academics in India at that point were not paid enormously good salaries. That's been rectified a little bit. So I think that was pretty much the, upper limit of what he could afford given the frequency of his habit. Luckily, he's actually given up, that's the other really amazing thing he did, which was he completely quit, more recently. I think his daughter had a more significant influence than his spouse did. So, yeah.
That's fantastic. And how then? So it's already a big jump from to this personal tutoring program. How did you find the topic for your PhD in the U.S.?
Well, I have to credit another gentleman whom I've actually never met in person, which is Mr. Richard Dawkins, because he published this series of books. But the one that actually had the most profound impact on me was The Selfish Gene, because at the same time as I was reading about the fundamentals of cell biology and molecular biology, I was reading about selfishness in biology, and those two views were so completely contrasting with each other, where one view was this perfectly engineered, Swiss watch, like, interlocking gears, everything works together, and it needs to for the cell and the organism to survive. And on the other hand, you had all of these genes that were basically hijacking the rest of the genome and the cell for their own self-interest. And I was just so struck by this complete contrast, opposite sort of themes. One was of complete cooperation and one was of complete conflict. And I just thought that, it's actually really wonderful to look at this topic and consider that there might be a way to reconcile this if you actually just look a lot closer. And that became, I didn't think about this as a elaborate sort of theme for myself, but that became effectively the driving force for most of my career, including my topic of choice in my PhD, where I was very interested in mobile genetic elements. These are elements like transposable elements that jump around in the genome and create havoc, but they do so because they are still increasing their own copy number in the genome. So the genome is their ecosystem. And I realized that the mobile elements provided a really good opportunity for me to look both at the evolutionary dynamics of these selfish elements, but also at the molecular biology of their mechanism. So I basically, completely like a naive student, applied to all of the big names in transposable element biology from India, having had no background and really no biology classes on my transcript. And luckily for me, there were at least a couple of people who decided to take a chance.
A small parenthesis here. I'm curious because there used to be this tradition, as you're mentioning, people would name books that really drove them to their field of research, going all the way back to the microbe hunters in the 20s. You're now a mentor, and you've been a mentor for quite some time. Do you still encounter that? And if so, what books are people referring to nowadays?
Yes, I actually think that tradition is completely alive and well. And we used to have the situation where a lot of the books were primarily written by academics who were in the field. I mean, Richard Dawkins is an academic of some repute. But what's actually a new tradition, which actually has, really blossomed, is we have professional science writers, gifted writers like Ed Yong and Carl Zimmer and others. So in addition to the academics who are beautiful and persuasive in their writing, we have people who are just gifted with words who write about topics related to science. And I actually think that, the sobering reminder is that no matter how many papers you publish and, the number of scientific discoveries you make, the actual impact that it'll have in a larger sense of a really good science book is, there's no comparison. A good science book is going to actually have a much more profound impact on educating the public and inspiring the next generation of scientists. In my own life, even though this was not related to biology, the TV show Cosmos by Carl Sagan had such a profound impact. There was an entire generation of people like me growing up at the time who became enamored with the concept of a career in science. And that was completely inspired by a physicist talking about the mysteries of the universe. So I think this is a very underestimated thing, and it hasn't gone away. Actually, the internet has probably made it a little hard to discover the true gems because there's so much stuff out there. But on the other hand, if you're a genuinely curious student, there is a lot that is available.
I feel obligated to ask, given this sort of passionate defense of the book. And at one point on Twitter, you joked about your editor. I don't know or agent. I don't know which one. Are you working on a book?
I am actually working on a book, but my ultimate plan was actually to devote most of last year to writing chapters of a book about ancient viruses in our genome, etc. However, the best laid plans were like way late. And ironically, I thought I would have more time to work on the book, but there were so many small and moderately sized fires to put out that I mostly just focused on the mental well-being of my family, myself, and my lab, more so than taking some time. I mean, writing a book is you might think that, as you know, like you're an excellent writer yourself. So it's not the kind of thing which you can do on a kind of a part-time basis. It really requires some time to really get in. And unlike others who are gifted enough that their first draft reads like poetry, for me, it is something that requires work. Like I need to chisel away at it multiple times and get better. And there are definitely times when it's very frustrating. So it helps to devote some time to it. And some of my academic colleagues who are much more organized than me have been able to do so. I mean, I'm just impressed by how much they're able to drive their own science as well as, write amazing books. I was not able to do that. And I'm hopeful that I will be able to do that next year.
I hope you do. I mean, I know a lot of people be looking very much forward to that. So getting back now to our main line here. So then you were working on retrotransposons, and after that, suddenly we see you working on centromeres, and out comes this really interesting concept that somehow brings the selfish gene sort of preoccupation and centromeres together, which is, I think, what you termed centromeric drive at one point. Could you tell us a little bit about that?
Yes, I'd be happy to do that. Actually, towards the end of my PhD, what was kind of a transformative moment for me was like stumbling upon the sort of field of transposable elements, even though it was a naive entry. It came at a time when a lot of sequence databases were being developed. And so I became very interested in the relationships between these selfish elements, how they evolved, what was their origins. And that's primarily what I focused on during my PhD. And I became more and more interested in deep homology searches. How far back could you use? And now, with tools like AlphaFold and SciBlast, it's becoming easier to peer further and further back, billions of years of protein evolution that you can go back. At the time, it was not so easy. You had to have your own tools. And I became interested in the lab of Steve Henikoff because he had developed these remote homology detecting programs, the blocks database, for example. Except when I interviewed in his lab, he was very interested in having me interview, but he told me he was getting out of that business because NCBI had become so good that he didn't really feel that he had something super original to contribute there. So here I was, I had traveled across the country. I was really excited about working with Steve Henikoff because even a 1 hour conversation with him just convinces you that this is just a amazing force of nature. But he was not going to be working on the topic that I was actually interested in working on. But he told me about these other topics that he had going on. And then he told me about centromeres. And this is a complete coincidence, which I still to this day cannot believe it was such good luck on my part, where he told me about this project that he had worked on where these histone proteins that are completely essential for chromosome segregation, the so-called centromeric histones, were basically evolving very unusually fast for a histone molecule. Histones are actually so well conserved that the amoeba histone and the human histone are only 2 amino acids apart. I mean, they're basically essentially frozen in evolution. Whereas the centromeric histones were evolving so fast that you could barely align them in many cases across different lineages. And then I had just read this paper from Chuck Langley's lab, which had talked about the potential conflict between chromosomes during female meiosis and during the formation of eggs, where, out of the 4 products of meiosis, only one of those products is destined to be transmitted to the next generation. And it totally clicked for me. And I can totally remember this because this was such a transformative moment in my professional life. We were having dinner after my first day of interviews at this Turkish restaurant. It was Steve Henikoff and his wife, Georgia, who's an amazing computational biologist. And before we knew it, the restaurant was closing because we had just been locked into discussion of this paper, and Steve's observation. And I was getting more and more convinced this was the explanation and we could test this, and it would actually also explain why the centromeric histones were rapidly evolving, but also the bigger paradox, which is why centromeric DNA, which is actually completely essential, so rapidly evolving. Base pair for base pair is the most rapidly evolving parts of our genomes. And nobody really said this in an overt way, but at the end of that dinner, I had another day of interviews in his lab. But I think I knew and Steve knew that I was going to come to this lab because this was just this was such an organic moment. And, I just feel so lucky that even though it was completely driven by like complete circumstance, and serendipity. And this was not the project that I'd applied to work with Steve for. I was just so lucky that this project and even my ability to contribute to that project had come about because of completely random reading.
It's interesting. At one point, you referred to it in another interview as one of the potential answers to Darwin's great mystery, right? So, well, as many people have noted over the last 100-something years, the origin of species never really explains the origin of species. Could you tell us a little bit about that?
Yes, I mean, I think there are certainly many observations, but I think one of the mysteries of Darwin was, if you want to increase the reproductive fitness, why would you ever create barriers between you and members of your population? And the transformative idea actually came from two papers, one from Steve Frank and the other from Lawrence Hurst and Paul Makowski, which they basically pointed out that perhaps selfish elements that are interested in their propagation could actually artificially lead to the situation where you have these boundaries that get created. And of course, what I was proposing was, well, you could actually have selfish elements for something completely fundamental. These are not rogue mercenaries in the genome, but even fundamental components of cell biology, like the centromeres, could be acting as selfish elements. And suddenly, basically, I took these two ideas, one, which was like completely coming from Frank and Hurst and Paul Makowski, and then the other coming from our own work on centromeres being potentially selfish, to recognize the cell cannot escape. This is just an inherent part of the biology where something completely fundamental could be itself responsible for creating reproductive barriers. In a way, again, the timing was good because when Frank and Hurst and Paul Makowski had proposed their conflict-driven speciation model, it was definitely not a popular model among evolutionary biologists because they felt that it had to be a lot more complex, etc. But now I think the field believes that whether the conflict is between the nuclear genome and the mitochondrial genome, or between different nuclear genome, or like in our case with centromeres and heterochromatin, that is a universal theme, that conflict-driven biology or conflict between different genetic entities is really at the heart of creating some of these reproductive barriers.
I mean, it's amazing, given how this field has evolved and how so much of it actually due to great work from you and your colleagues. When you look at the diversity of different conflicting genetic elements, it looks miraculous that there is a cohesive or semi-cohesive genome at all. It really looks like one giant centrifugal force going all the time, every little base pair for itself almost.
Yeah, I mean, I can make the analogy between like a settler population, right? Like which has to band together to fight off all of these mercenaries and bandits that are constantly invading the village. It's in the best interest of the villagers to band together. Otherwise, they're going to be taken apart one by one. So I sort of view in a sort of old Western or old kind of Kurosawa film kind of way that the genome is really divided into the bandits and the people who are fighting the bandits. And that occasionally the bandits become part of the village and occasionally the villagers become bandits themselves, which makes life a lot more interesting.
Well, and sometimes you have, exactly that, becoming part of the village side of the story, which is super interesting when the bandit is now the baker in the village, when you get a placenta out of it or something.
Exactly.
It's incredible. I mean, just how far this has come in the last 30 years. And to come to one of our topics, to review commons in a very circuitous way, I saw one of your recent presentations online at the Hutch where you're talking about where you're displaying this large discontinuity in the number of active retroelements and retroviruses in the human genome versus the rest of even primates, even the larger primate family. And this was so unexpected, right? This is largely what tanked the NCI cancer program in the 70s, was you thought we were going to go from patent rouse to the retroviruses that cause cancer, and that would be that. And as a side effect, we got all of the HIV research field and all of HIV therapy. So it's not so bad, but still, this was a giant unexpected thing between the chicken that was brought to Rockefeller and the realization by the time that in the early 80s that you have HTLV 1, 2, 3, sort of, not actually. It's HIV, but it's incredibly poor in the human physiology active endogenous retroviruses. Why is that?
I think it could be a number of different questions. I do think that the effective population size of the host plays a very key role in terms of vertical and horizontal transmission of these elements. And I think the nature of the dispersed populations that we've gone through, or our ancestors have gone through, probably played a key role in the extinction of a number of these lineages as it kind of happened. I mean, a very similar argument can be made about why some viral pathogens become less virulent. You know, there's a debate going on right now because of SARS-CoV-2. Is that on its way to becoming less virulent? And there's actually really good theory work that people just fail to cite all the time, which is criminal given how high quality it is, where really it's not even a viral determinant. Actually, the key determinant of whether a virus is going to evolve a virulence is whether it's in a population where it has ready access to new hosts, or it runs the risk because of this hypervirulence of extinguishing the host population that it absolutely needs for its own propagation. And the good news or the bad news, as you might see it, is that, you know, we live in an age where a human adapted virus has got no dearth of new hosts to kind of interact with. And so this is a little bit of a, you know, yes, this is going to become an endemic virus, but to predict that this is definitely on its way to a virulence actually just reflects a poor knowledge, or perhaps a little bit more of a rosy view of what actually happens with virus evolution.
Well, and I guess there are several other factors. One is people forget the time scale to putative endemicity is great, but if it works from the evolutionary point of view, not so good looking at the history of recent human viruses.
Yeah, I would also just like to make the point, you know, it's interesting that we have so many people who are suddenly believers in Darwinian evolution because we're seeing this in real time. But the thing that people forget is that Darwinian evolution is very bloody. You know, the fact that you get selection for 1% of the population, it's because 99% of the population was basically killed off by the pathogen. And that's a very high cost. And when you put it in those terms, you sort of recognize, wow, we're really talking about a pretty high burden here, which is something that I wish that more public health and epidemiology people would like incorporate into their thinking. You know, here's the case where the evolutionary biologist has got more humanist tendencies than the public health professionals.
Well, and somehow in the middle of all this, endemic also became a benign word. So HIV is endemic to equatorial Africa. Plasmodium is endemic. I mean, trypanosomes are endemic. None of these are good things to have. And, you know, if you pull out the antiretrovirus, you'll see very quickly just how benign endemic HIV is in equatorial Africa. So
yeah, I refer to it as like a negotiated truce. I mean, herpes viruses in our own genome are a perfect example of that, right? We've kind of co-evolved with them for hundreds of millions of years. And, you know, they're trying their best not to like hurt their host very much. But if you get into a situation where you're even transiently immunocompromised, like we see in our, you know, institution with people who are going through cancer therapy, wow, they come raging back, and they're not really good, you know, viruses to have coming back in a big way. So these are not viruses that have like defanged themselves. They have basically been driven to this impasse because of the immune system that we've basically come up with. And it's a constant battle. It's not the thing where the battle is over. The battle has just reached this impasse where both of them are at their, respective boundaries, just waiting for any sign of weakness on the other side.
You've sent so many great manuscripts to Review Commons. One of them is this Applebeck 3 paper. It reminds me a bit of something you were saying earlier in the conversation about not just domesticating things, but sometimes using the tools of the potential pathogens or the selfish genetic elements as a defense against them, right? Could you tell us a little bit about this paper?
Yes, so I'd be very happy to. So this is a paper that was actually sort of a joint product of my lab and a recently graduated postdoc, Rick McLaughlin's lab. And we were very interested in APOBEC proteins. APOBECs, for people who don't know, are these cytidine deaminases that target retroviruses and transposable elements during a stage in their life cycle where they're single-stranded DNA. And they introduce mutations. And if you introduce enough mutations in that genome, you can basically render the virus essentially ineffective. So you can kill the virus by poisoning its genetic lineage, if you will. And what we discovered was that, yes, there are these APOBEC proteins that are in the genome, and we think about them as the normal intron-carrying APOBEC proteins. But there were also these additional sort of idiosyncratic APOBECs that we were finding in the genome that lacked introns. And, since we know that the ancestor was intron-bearing and the daughter gene, if you will, were intron lacking, we know that this must be the product of reverse transcription itself, where the APOBEC gene was first converted into RNA, and that RNA was then reverse transcribed and integrated in the genome. And what we basically found was that there was this actually incredible proliferation, not just once, but throughout, and it's actually ongoing in many primates, including in marmosets, where APOBEC repertoires are constantly expanding. It's almost as if the genome has taken advantage of this resident selfish element, the line 1 retrotransposon, and is actually using it to almost as a homeostatic mechanism, increase the amount of APOBEC diversity that it has to defend against the very retrotransposons that gave birth to the retrogene in the first place. So it's almost as if it's got this pressure cooker-like system. If you've got a lot of retrotransposons that are unchecked, you're going to make a lot more APOBEC retrogenes. And then once there is a lot of APOBEC retrogenes, they will quiet down the retrotransposons a little. So it's this almost like beautiful inbuilt homeostatic mechanism in many genomes from our closest relatives.
It's got an interesting parallel in the adaptive immune system, where the most accepted model is that we have antibodies and T cells because of a diversification mechanism that is not a deaminase, but a recombinase that was also domesticated, probably retrotransposon. And one of the things I wanted to go over with you, because in a recent event with the Whitehead Postdoc Association, you'd said that one of the things you appreciated about this model of review commons, where you submit, you get the reviews, and then you say what you will or will not do to the editors downstream, right? And that saves time, that avoids confusion. And I was looking through the files of this paper, which is on eLife, and it has the review files at the bottom. I think everyone should always have a look at these when they're available, because they're a great tutorial, not just for the science, but also for people who want to learn how to peer review. And one of those cases here was a reviewer, I think number one, was saying, well, great, these are great predicted structures, but can you do functional assays for 20 of them? And what happened after that?
Well, in a way, multiple things happened at the timing of the review. Like, it was our first experience with review comments. I was really excited by the idea, but then also, we were unable to do many of these experiments because we were not permitted to be in the lab. And different journals took, I would say, a graded view of how sanguine they are with what is really critical. And what was lucky here was I think the reviewers were quite good in the sense that they looked at both the positive and the sort of what the missing pieces in the work. We actually looked at those missing pieces, and we ourselves actually parsed into, okay, some of these missing pieces are absolutely critical for our story to be complete. Like, there were holes, and admittedly, there were holes in terms of making sure that the logic was absolutely crystal clear. There were other things that would be really nice to have, but given that we had such a limited amount of time that we could go back into the lab, we basically parsed it into, we will do these things. Some of these things can be done computationally, and then the limited amount that we needed to do with wet lab access, we could do. But we basically did that ahead of time. So rather than doing like a revision plan, we said, look, we don't want to submit this until we've taken care of this. And then we submitted this, and we said, look, if the editor feels that this additional work, which we felt was was good, but not necessarily relevant to the main thesis of a paper. If the editor felt like it was necessary, we would do this. But we made the case that we don't necessarily think that this is necessary for the main thesis of our paper. And luckily for us, the editor, at the time at eLife, they felt that, yes, I think you've made your case. I think you've done the work that we feel you need to do to have made, your case a lot more airtight. And yes, while these are really excellent additional experiments, they could easily be a follow-up paper rather than something that needs to belong. That APOBEC paper is a really good example, but there are even other really excellent examples where there was, the reviewers were not necessarily all like thinking the same. And so we had like a lot of reviewers. They're very constructive, but, if you had done all of that work, that would be like almost 2 papers worth. So there we basically said, we're going to come up with a revision plan, and this is something that we will do. And this is something that we will do if the editor feels it's completely necessary. And we sent that, again, as it turns out, to eLife. And there again, it was really great because the editor essentially looked at our proposed revisions, which we had not done, and they agreed with all of them, and they added one of the what we considered potentially optional ones. They said, you should do this because we think this is really important. And having that in hand is such a great sort of thing because, there's not uncertainty. There's not a changing, the most frustrating thing about some of this process is that you do your work, you do your due diligence with reviews, you go back, and now you've got either a new set of reviewers or a reviewer who says, this is great, but how about you do this additional thing? And then you sort of realize that it's a red queen, like you're constantly chasing this myth of like the perfect paper, which, it's a continuum. And so having the certainty, as I said, in this event that we had with the Whitehead Postdoc Association, is almost the best part of this process, is that, whether it's a refereed preprint or it's this conversation that you've already had with the editor about what is needed for this paper to meet whatever that bar is for acceptance. You're working with that sort of certainty in mind, and you may not be able to meet the bar. And we certainly were not able to for one paper for completely technical reasons. We could not do it. And even then it's good because now it's clear like the editor does want this. And so if we can't do this, we will basically adjust our sort of sites accordingly. And so not just for cases in which, the paper eventually got accepted, we've had, I think Thomas can correct me, but we've had, I think 9 papers go through review commons now, and 8 of the 9 were accepted in the first, journal that we actually submitted to. One of them was not, but the one that was not is almost the best advertisement for why Review Commons is so good, because that one was rejected within 24 hours, and we were given an option of another really excellent journal, which did not require this experiment, which we had basically failed to do for technical reasons. So again, there's certainty because in a 48-hour period, we submitted our paper, got it rejected, had a conversation with the editor of the second journal who was on board with our revision plan. And now we had like a path forward. And that is worth its weight in gold. I can tell you, as if you talk to a practicing academician who's already invested, many man years of work into a project, having that certainty, this is the path towards success and publication is really great. And I think in a way, the exception really proves the worth of something like Review Commons.
Review Commons project leader Thomas Lemberger asked Harmit to tell us a little bit more about his views on the interplay or tension between peer review for scientific quality and journal fit.
I'm curious. You mentioned, this concept of you are not sure that the paper will meet the bar of a given journal, and you mentioned this concept of a revision plan where the authors can say, I intend to do this, and these are experiments that I am not able to do, or maybe also I disagree that they are absolutely essential for my conclusions. And we have a lot of discussions internally whether the sort of journal agnostic philosophy of the peer review at Review Commons how pure and how fanatic we should keep with that principle, and whether it might be an advantage that either the authors or the reviewers would provide suggestions of which journal might be suitable for a given manuscript, or maybe the authors could pitch their manuscript for a range of journals, and then the reviewers could comment as a function of this pitch, or whether we should really keep to our ethos of journal agnostic and really separate the work on the science from the peer reviewers. And then the editors of the journals have this function of curation, editorial selection, aggregation, and essentially having this dialogue with the authors on what is the bar of that specific journal. So where would you stand? Would you stay pure, or should we sort of have a little bit more of a compromise?
Yeah, it's an excellent question, Thomas. Obviously, this is a little bit above my pay grade, but I can give you the perspective of an author that the journal agnostic process has been one of the best features to us as a submitter for that, because essentially, the problem is that if you can be guaranteed that the reviews and the comments you're going to get are going to be the same, whether this paper was targeted to EMBO or eLife or PLOS Biology versus PLOS One, then I think it's a problem. The problem is that the whole problem with the review system, which is sort of journal driven, is once you start reviewing a paper for, let's say, Cell or Nature or Science, you've automatically formed an impression in your head about what kind of paper appears in these journals. And now you're not just evaluating the paper that you have, you're actually evaluating it against your subjective impression of what needs to be in the paper. And what was actually great in many of our cases is that we were basically evaluated by experts that told us about what they felt, and it's not like they were always uniformly complimentary. In some cases, they basically said, "Look, this has the makings of a great paper, but there are these really important controls that you have not done." So it's not that they were like pushovers in any respect, but they were really focused on our story and what we needed to do to make it complete, rather than this sort of pretty arbitrary bar. And the nice thing about that is that review process and those sets of reviews are static. Now the editorial look at whether that's actually good enough for their journal, that process is short because the reviews are already there and we can port those to multiple journals if needed. The problem with the journal sort of non-agnostic view is that the editorial process and the review process are intertwined and often need to be restarted because it's hard to get over the bias that the reviewers had in the first place because they were reviewing for what they think is a quote-unquote high-impact journal, and they're basically measuring against that artificial bar rather than actually just measuring the work as is in front place. And that's one of the best things about preprint and bioarchives as well, is that you think what you think is your best work in its form of completion and you put it out for the world to see, because you're already willing to stake your reputation by putting that paper out for everybody. And in a way, you're doing the same thing when you submit it to a journal. But I totally understand there is, whether we like it or not, there is a hierarchy in the journal in terms of desirability. The problem is that desirability then actually changes not just the subjective opinion of the reviewer, but sometimes what they feel is the objective opinion of the reviewer. So it's actually, it's kind of, for me, it can be hard to remove that subjective opinion from the review process. So I really love the journal agnostic view. I'm an editor on a number of journals myself, and I do see cases where this paper, and I have to step in because, they are sort of even making this explicit or implicit judgment of, is this paper good enough for this journal, rather than, is this paper really saying something cool and new and novel? And many editors are not willing to do that because it's too much trouble to go against the reviewer's subjective opinion. Yeah. So I love that feature that it's journal agnostic right now. I think it may not be completely out of the question to recommend reviewers to suggest journals, and they can do that either online or offline. But there again, I think the reviewer's own subjectivity about what their impression of this journal is will creep in. And reviewers, authors tend to be completely unconservative where they want their papers to appear for good reason. Reviewers actually tend to be on the other end of the spectrum. They tend to be a little bit more conservative. Unless you get somebody who's just totally blown away by the novelty of the paper, they tend to maybe underestimate what the paper might be, even though they think objectively the paper is very good, but it's a subjective opinion about which journal it should actually appear in.
Sure. So you mentioned, posting a preprint is a big step, right? You reveal your paper, your science, your research, your results, potential mistakes in the public, and it takes a little bit of courage, right? It's an important step. And with Review Commons, we also provide the opportunity to post the reviews then next to the preprint. So what has been your experience, and maybe also of the younger people in your lab? Are they comfortable doing that, or do they hesitate a lot? What's your feeling?
Yes, so this is where there is definitely a lot of distribution of opinions. So, for example, of the 9 papers that we submitted to Review Commons, we had already posted preprints on bioRxiv for 8 of them. For one of them, we didn't do that because I was not able to convince the first author that the pros outweighed the cons, and it can happen particularly in fields which are really fast moving and you're really kind of worried. Ironically, actually, in those fields, it makes more sense to post preprints because they're fast moving enough that you get a timestamp based on when you put your paper up, things related to CRISPR-Cas9 based engineering, etc. We are seeing more and more examples of very different groups that come up with very similar conclusions and are often putting preprints, within a day of each other. So I think there are very few cons related to putting reviews up. There have been a few cases where we've delayed putting the reviews, mostly because, as I said, there were actually instances where something pretty fundamental in terms of a control had not been done according to the reviewer to the best quality possible. And there we felt like, it is actually possible if we did the controls as the review wanted, a fundamental aspect of the paper could change. And so we felt like we needed to do at least that experiment before we felt comfortable sharing everything. In a way,
that's interesting.
The review process is as much about asking your best, the best opinions from people who you think are the experts in the field. And if that's what I mean about parsing, like if we feel like this is really fundamentally critical to the validity of our argument, I feel more comfortable having done those experiments and before I put it out, because I also don't want to mislead, my colleagues.
It's no interest to anybody to have possibly wrong claims, and
exactly,
and prefer to revise the paper and have a solid thing to.
Well, in each case, it turns out that we, even though we had not done the perfect control, when we did do the perfect control, it was fine. But I also felt like I needed to be in a situation where we didn't need to backtrack a lot of things, because then it gets very difficult. So that's been the thing where we don't put it up instantly, but we basically, and I also feel really comfortable putting a revision plan. And if that's there, I'm happy to share it also. Some people are more interested in making that conversation as private as possible. I can understand that because they're trying to not influence subsequent editors who will see this. I just feel like I want to make sure that we are on very solid ground. And then, as I said, there's always going to be optional experiments. And I'm okay with the world knowing that, because they're actually often experiments we love to do after the paper is already published, because it's, as I said, everything is a continuum. It's not like the paper is the last word on that particular topic.
So I'm sure that you serve very frequently as a reviewer, and of course you receive a lot of reviews on your own manuscripts. So peer reviewers, they have a very tough job, right? On one hand, they want to be as constructive as possible. They really want to help the authors to make the paper better. And so they want to be the nice guys. On the other hand, they also have to be they have this sort of formal role to see if the evidence, if the reasoning is correct, if there is a flaw. And so they have really to dig and to kind of challenge, right? This is part of science, is to challenge and see, is that really true? So what's your feeling about being, the nice dude and being the rigorous, analytical guy who is very strict? So how do you combine those two aspects?
So this is a great question. I actually think that those are not as opposed to each other, Thomas, as you think, because in both cases, I feel that my job as a reviewer, whether I identify myself or not, is to make sure that my colleagues who've written this paper have not made a mistake that they will regret later. It's fine if you get your paper into a really high profile journal, but if 2 years later somebody discovers a fundamental flaw in that paper, all of the goodwill you earned with writing this really beautiful high profile paper is completely, it becomes a complete negative. So I want to make sure that, just like I would do for a colleague down the hall from me when I'm reading their paper, I want to make sure that I give them constructive feedback, but I also want to make sure that the paper as it stands is solid enough that it'll withstand the test of time. And I sort of don't view those as being adversarial roles. Where it becomes adversarial is actually where you then evaluate the totality of the paper in context of like how important is it to the field, and then you make a subjective judgment about, is this innovative enough? Does this meet this bar that this journal has set for impact? That's a completely subjective opinion. Some people are a lot more difficult to please. Others are easier to please. Ironically, and perhaps unsurprisingly, journals like to go after the people who are going to be hard to please because they can only publish, a very small subset of the papers that they get. But also, people can take a view that, this is a great paper in my field, and it will bring attention to my field, so I should be more encouraging. So that's a self-interest driven thing. Or ironically, well, there's only going to be one paper in my field published in a high-profile journal. Why should it be this paper and why not my paper? And that's a self-interest in the opposite direction. So I'm just saying that the decision about whether the paper is accurate does not have to be an adversarial decision, but the decision of whether the paper is appropriate for this journal, there's inherent bias in that. There's no way to actually get around that, which is why I like the bioarchive and the review commons model, which is that we're going to evaluate the paper as it is without sort of thinking about where they should eventually end up.
I mean, in a way, we try really to help also the journals making that decision by asking the reviewers, to comment about what we say, the significance, what kind of advance has been made. Is it a methodological advance? Is it a conceptual advance? Is it, degree of coverage? How does it compare to the existing literature? In a way, we try to sort of find, a path to for the reviewers to describe the significance in as objective way as possible. Of course, it remains slightly subjective, with regard to the background of the reviewer. But we try to provide a little bit of a framework how to express that, even if it's just focused on the science. Do you think this is working? Do you think that editors have enough information and to make their call? Is that helping the judgment?
I think it is. I can speak both as an author, but also I've received papers that have been transferred, for example, to PLOS Genetics from Review Commons, and in 2 out of 3 instances, the paper was completely acceptable as is. The authors had made a few revisions, and we spent 2 days at the journal before the paper was accepted. In one case, the authors, had to do a lot more, so they came up with a revision plan. And, in 2 to 3 days, we said the revision plan is acceptable. And then a couple of months later, the paper was accepted. Again, I think it's just giving people like this path to things. I mean, I can tell you as an author, we've taken snippets of those significant statements that the reviewers have written, and as part of our pitch to the cover letters to the journals as well. I mean, I'll just tell you though that there have been cases, so the decision of whether to send it to Review Commons or to send it directly, let's say to eLife or PLOS Biology, there have been papers that we've actually sent to eLife, which were not reviewed or editorially rejected. And completely to my surprise, in fact, I think like literally one of the best papers the lab has published. So surprising that I never do this, but I challenged the decision. I said, like, are you really sure? I mean, this is super important, but and the editor is extremely good. It's not like they're not, aware, but they're looking at the paper from what it doesn't have rather than what it has. So we said, okay, let's send it to Review Commons. I mean, at Review Commons, it had like glorious reviews, right? Like there were 4 reviews. They were absolutely glorious. They focused again. They did also have some of the same inquiries that the editor had about what we had not done, but they actually also said what we had done. And I am 100% confident that if we had those reviews and had gone to eLife with them, we would have been accepted. But, and there's no harm. I mean, the paper ended up in a fantastic journal eventually anyway, but it just tells you that it's always good to not be wearing that hat of like, oh, is this an eLife paper? I think it's really good to evaluate it like, okay, what's the paper? And then say, wow, this sounds like an eLife paper. And that's the process which having the scrutiny being more detailed in the beginning rather than at the end, it changes the process. This is what I'm saying. It's not the same process anymore. You cannot give the same detailed scrutiny that you would with 4 really detailed reviews in hand. And that's where I think Review Commons, I mean, honestly, don't see any reason why we would be sending papers to any place else because of the advantage it actually puts in the author's bag. It is in your advantage to actually have. And, the thing is, if you get reviews that you don't like, fine. You can, pull your, slot wheel or the roulette wheel or whatever your favorite gambling analogy might be and go to the journal directly. But I think it actually gives you the best odds of getting your paper evaluated fairly. And that's the best thing that you can actually want in the process.
Well, it was great pleasure to have you, Harmit. It was really great fun talking to you. Thank you so much.
My pleasure. And thank you again to the team as well at Review Commons. It's really made a big impact. Thank you again, Tiago, for taking me down memory lane.
You can explore more of the Malik Lab's science at research.fredhutch.org. To find out more about refereed preprints, you can go to reviewcommons.org. Thank you for listening to the EMBO podcast.